bottom of the question that the diameter is increased?
Yes. So positive would mean that it decreases in diameter, negative would mean that it decreases. This is the okay, just a couple more minutes. Okay, so let's go over the answers to this. Okay, so do you think rennet increases or decreases blood pressure? Increases. And why does it increase? What does it do to the blood vessels? Makes them constrict. So rennet actually is eventually causes constriction of the blood vessels. It also causes the release of aldosterone and ADH, which both increase blood volume. So you could put, it increases by actually causing constriction, okay? So ADH release, okay, so that increases the reabsorption of water by the kidney. Remember, the kidney also releases rennet. So that would increase or decrease. It's increasing blood volume, it increases blood pressure, okay? How about if you take a diuretic? Decrease blood pressure. And so there's some things that we sometimes normally consume. So alcohol can be a diuretic. And so you have to urinate a lot when you drink alcohol, and you also tend to get dehydrated because not only are you getting rid of the alcohol, but it takes a lot of water to get rid of the alcohol as well. And so that's why they say that you should continually drink water if you're drinking, right? So if you have a beer, then after every beer or between, you need to drink a bunch of water. And so the effects of being hungover generally are just the effects of being dehydrated. And you might have heard that some people pay a nurse to come to their house when they're hungover. And what does the nurse do? Right, so it actually gives them water intravenously, right? So they actually give them an IV to rehydrate them. It's kind of interesting. Whenever I've gotten an IV, I feel so hydrated. It's like, I don't know if you've ever experienced that, but it's just like you feel like it's just amazing that you're not thirsty because you haven't drank for a long time. You just aren't thirsty because you feel so hydrated from the IV. Okay, so vasodilation would decrease. Release of aldosterone actually increases. Okay, so aldosterone causes sodium ions to be absorbed by the kidney, and when sodium ions are reabsorbed, water moves with them, so that increases blood pressure. The increase in the blood viscosity, increase, right? So that affects resistance, so you have more peripheral resistance. Vasoconstriction, increase. How about the release of ANP? And remember, that's produced by the heart. Decrease, because that is a vasodilator. So that's a hormone that's actually produced by the heart. Relaxation, decrease. How about smoking? Increase. So smoking not only increases heart rate, but it also is a vasoconstrictor. So nicotine that you get from smoking constricts the blood vessel as well as elevates the heart rate. And then atherosclerosis, increase, because it causes an abnormal narrowing of blood vessels due to the deposition of plaque in the blood vessels. Okay, so then we have arterial vessel diameter. So how about the presence of nitric oxide gas? Increases, because that's a vasodilator, right? So that would be positive. It increases the diameter. Low O2 concentration, increase, right? So let's say, for example, if your muscles are using lots of oxygen, there would be, and it's metabolically active, then we want more blood to flow to the muscles to deliver oxygen and nutrients. So it is a vasodilator, so it would increase. High blood pH. And this actually goes hand in hand with the high levels of CO2. So carbon dioxide, when it's dissolved in water, produces carbonic acid. So it makes your blood actually more acidic. So it causes respiratory acidosis. And so that would be a higher blood pH, and high levels of CO2 would both be related to metabolically active tissues. And so those would both dilate blood vessels, so more blood goes to them. Okay. ANP, we just mentioned that's a powerful dilator, right? Vasodilator. Okay. How about fewer impulses from the sympathetic nervous division? Increase. Okay. So remember that I mentioned that the sympathetic nervous system is the only part of the autonomic nervous system that innervates blood vessels. And so it actually is also the only part of the autonomic nervous system that innervates sweat glands. And so if you get a lot of action potentials being generated, then it constricts it more. And if there's just a few action potentials being sent to that smooth muscle, it will dilate it. Okay. How about stretching of blood vessels by blood flow? Decrease, right? So that's called myogenic mechanism. And so that's the idea, is that when you stretch, specifically in those conducting vessels and the arteries where there's lots of smooth muscle and lots of elastic tissue, like the brachial artery, for example, when you get that big push of blood through because of ventricular systole, then the blood vessels will contract against that blood pressure. Okay? And high levels of CO2 would be a dilator, so that would increase the blood vessel diameter. Okay? So this will be helpful for when studying for the final exam, right? Because you can see how I could make pretty easy multiple choice questions based upon these things.
How many questions are there?
I don't know yet. Usually it's like 50 multiple choice. That might be more. Sure. I haven't written it yet. Okay, so we were talking about the functions of the lymphatic system, and I gave you a handout, and we're going to need that handout again because we're going to look at the different organs found in the lymphatic system. Uh-oh, it's freaking out on me. What's going on? Okay, so we were talking about the lymphatic tissue, and remember that I mentioned that it's reticular, a lot of it is reticular connective tissue that is filled with white blood cells. And so the types of white blood cells include, and you should have this in your notes, the macrophages. So macrophages are large phagocytes, but these also include the lymphocytes. Right, so we have lymphocytes. Ugh. Okay, so are lymphocytes granulocytes or agranulocytes? Does anybody remember? Do they have granules in them or do they lack granules? They are agranulocytes. So they, along with monocytes, do not have granules. So you would say that these are agranulocytes. So remember that all of the white blood cells can be categorized as either having granules or not having granules. Macrophages do have granules, and that's because they have vesicles filled with enzymes, and that's what makes them look granular underneath the dissecting microscope. Okay, so these are agranulocytes, and there's two different kinds. And so we have the B cells, and these mature in the bone marrow. So they're produced in the bone marrow. All blood cells are produced in the bone marrow, but they mature in the bone marrow. Okay. These are specifically designed, if you remember when we were talking about the leukocytes, these are what undergo division, and produce cells that specialize in producing antibodies. So these produce antibodies in response to foreign antigens. And these antigens could be pieces of microbes that are actually presented to the B cells by the macrophages. So macrophages eat bacteria, and then they present the antigens to the B cells. And so lymphocytes, including B cells, reside in greater numbers in the lymphatic tissue, like in the lymph nodes. Okay. So we have another type of lymphocyte, which is called the T cell. And these are produced in the bone marrow but mature in the thymus gland. So the thymus gland is up near the neck. So if you think about it, your thyroid gland is like in the middle, and then your thymus gland is on either side. And in babies and in young people, it's so big that it actually kind of extends down and comes into the thoracic cavity. But as you age, like after you get through puberty, your thymus gland will actually start to shrink, and it'll get smaller and smaller and smaller. And old people have very, very small thymus glands. And so oftentimes when we do the cadaver, we can't even find the thymus anymore because it's shrunk. Okay. Same thing with, we talked about tonsils. Tonsils, like, I have not had my tonsils out, but the dentist always thinks that I do because he can't see them. Like, they're almost completely gone. You can't find them, right? So your tonsils can shrink down when you get older, which is actually a problem because it means that your immune system isn't as strong. And it might be one of the reasons why, as you age, you are more likely to get pneumonia and you're more likely to get cancer. And those are two things that you can die from. Okay, so this is the thymus. So it's called the T cells because it's produced in the thymus, okay? Or they're not produced in the thymus, they mature in the thymus, okay? And so if we look at the diagram or the image that we saw, oops, I have my lymphocytes there. Oops, I thought I had my picture here, sorry. Must be beyond one back. Where is it? Oh, well, must have got erased. Okay, so there's a picture showing the reticular nature of the lymphatic tissue and then the presence of the lymphocytes. Okay, so we can talk about lymphoma. And so what do you think lymphoma is? Cancer, right? And so it's different than leukemia in that it is due to the cancer of the lymphocytes. Okay, so it's actually kind of a type of leukemia. Okay. And so remember that we talked about how if you were looking at whole blood and you were looking at the hematocrit, which part of your whole blood hematocrit would be enlarged? The buffy coat, right? So the buffy coat would actually be bigger because there would be more white blood cells, okay? So when we look at this, one of the things that happens is that we get swelling of the lymph nodes. And this is kind of an unusual cancer because it can be relatively common in young people. Like you can get people in their 20s, for example, getting lymphoma. So that's kind of a common cancer that young people can get, or it can also occur in old people. So when people come down with it in their 20s, it's oftentimes very treatable. And so it's not as dangerous as some other cancers. So we have two types. We have Hodgkin's, and this is named after a person. So we have Hodgkin's lymphoma. And this is actually the more rare. So about 10% of lymphomas are this. And these are characterized by abnormal B cells. And these B cells are also named after the people that discovered them. So they're called Reed-Sternberg cells. Okay, so those are the two last names of the scientists that discovered them. And they're huge. And so if you look at a smear of blood and you see these Reed-Sternberg cells, that is a characteristic of the Hodgkin's lymphoma, and it's specifically B cells. And it is also associated with being infected by the Epstein-Barr virus. Now, viruses can cause cancer because one of the things that viruses do is they inject their DNA into the host cell. And sometimes that DNA, the viral DNA, causes mutations, which then cause the cells, the normal host cells, to become cancerous. And so it could insert itself into an important gene that regulates cell division. Okay. So if we look at an image from a slide, okay. So these are normal lymphocytes. Lymphocytes are agranulocytes, but they have this huge nucleus. And so this is the nucleus here. And then this would be, they've kind of drawn around it, showing the demarcation, how big it is. But it just gets big. And so I imagine the reason why it gets big is that the genetic material replicates, and then the cells do not divide. And so you see those Reed-Sternberg cells. So the other 90% of the types of lymphoma are non-Hodgkin's lymphoma. So this is about 90%. Okay, and this involves both, or can involve both B and T cells. So one of the problems with having cancerous cells in your circulation, in your circulatory system, as well as in your lymphatic system, is that they can go anywhere in the body, right? So your lymphatic system's all over. And so that generally means that you have to use kind of a chemical chemotherapy in order to kind of catch it, because there's no way that you can isolate it and remove it because it's kind of everywhere in the body. I guess you could isolate some of the lymph nodes that are specifically infected with the cancerous cells. Okay, so that's lymphoma. Here I was looking for this picture. Okay, so this shows the lymphocytes, right, in the lymphatic tissue. These are the reticular parts of the reticular connective tissue, and then this is the macrophages. So macrophages eat bacteria, then present the antigens to the lymphocytes, and the lymphocytes can respond. The T cells can actually directly attack the pathogens or the cancerous cells, and the B cells produce antibodies that bind to the antigens. Okay, so if you look at your diagram, I wanted to show you this because I had this image here. So we talked about tonsils, right? And so we talked about the different types of tonsils. So we talked about the palatine tonsils. So the palatine is because they're near your soft palate and hard palate right here. And so this is what those tonsil discs look like. So a person looking in the mirror would see huge tonsils. And this is just, some food debris, but also it's an infection because it's kind of white and pussy. And so they can pop these out. And so those are the tonsil stones. Yes. Yeah. So they just come up. Yeah. So my daughter actually has these, and she coughs them up occasionally. Like when she's coughing and she's sick, she'll cough up her tonsil stones, and then her tonsils will bleed.
Are they painful?
Yes, because they are an infection. And so we're actually getting her tonsils out over spring break. So that'll be interesting because she's 15. They do fall out naturally, yeah. But she's had them for over a year, and they said that she's just kind of got a low-grade infection in her tonsils all the time, right? But she's at a higher risk for pneumonia if she has her tonsils removed. So that's kind of a trade-off between the two. Okay, so I already pointed out the tonsils, right? So this is the pharyngeal, this is the lingual, these are the two palatine. So what do you think this is right here in the middle? Thymus, right? So this is my thymus. Okay, and this is representing the bone marrow. So that's where the lymphocytes are originally produced and where the B cells mature. What do you think this structure right here is? Spleen. So the spleen is part of the lymphatic system, and we'll talk about that in a second. And then what is this structure right here? Appendix. And these little yellow dots right here, or they're not yellow on your page, but these are called Peyer's patches. And we're actually going to watch a little video about the Peyer's patches in a second. Okay, so those are the organs that are in the lymphatic system. One of the important things when we realize this is that this is a little bit separate from the lymphatic vessels. And so the lymphatic vessels actually don't penetrate the appendix. They don't actually penetrate the spleen or the thymus. They do eventually, remember, those lymph nodes actually deliver their content, the lymph, up to the circulatory system, and it goes into a vein, which is significant because it is under low pressure coming up, and it wouldn't be able to put it into an artery because the artery is under high pressure. So it goes back into the veins. Okay, so let's look at the spleen. Oops, oh, I wanted to point this out. So bubonic plague is a bacterial infection that is known as the Black Death. And so this was the back in the Middle Ages, like a third of all Europeans died of this disease. And so it's called bubonic because of the swollen lymph nodes, which are called buboles, right? And it's in the groin and the armpits. And these swell up and you get death of the tissue surrounding it. And so that's why it's called Black Death, because you get necrosis. So the skin and the tissues die, and then it starts to decay. So that's the bubonic plague, and that's a bacterial infection that's carried. Okay, so let's look at the spleen. So the spleen is not directly connected to lymphatic vessels. And unlike the lymph nodes filter the lymph. This filters the blood. Right? So there are lymphocytes and macrophages in the spleen. And one of the things that they break down or that they filter is the red blood cells. So we have those old red blood cells. So old erythrocytes. And remember that the red blood cells, because they lack a nucleus, don't have a very long lifespan. It's about 120 days. And so they actually go, the circulatory system delivers the blood to the spleen, and then the spleen is filled with reticular connective tissue that has white blood cells in it that filter out the old red blood cells. And this occurs in what is called the red pulp. And you can see it is a darker color histologically. Oops, not pulp, not blood pulp, sorry, red pulp. Red pulp. Okay. Okay, so it breaks them down, and it also breaks down hemoglobin and stores iron. And this is the same, also the same thing that the liver does, right? So it stores, the liver and the spleen store the iron. Right? And interestingly, it also stores oxygenated blood. And in us, it's a small amount of blood, but in some organisms, and actually in some humans, they actually store more blood so that they can hold their breath for long periods of time. So for example, when you look at the physiology of aquatic mammals, like seals, the reason why a seal can go down and dive down and stay under for like 5 to 10 minutes is that when it starts to run out of oxygen, the smooth muscle in the spleen will contract, and it will send oxygenated blood that has been stored in the spleen out to the brain and to the muscles. And so it's kind of interesting because we do see people that train, the free divers, that train to hold their breath for long periods of time underwater, like 5 minutes, I think is maybe the maximum. But they can hold their breath for long periods of time, and it's one of the things is that their spleen actually gets bigger to compensate for that. So they can hold more. This is a reserve, right? So this is a reserve, a blood reserve. Yep.
Are there any negative effects to this type of training and stuff, or is it just something that happens?
Well, you can die from it. You know. So people come up too fast, you know, they can go down to, you know, too deep, and then the pressures, and then sometimes they come up and the capillaries in their lungs will burst, and then they'll die. So people, if they don't train properly, they will die.
Would it happen from just like aerobic exercise, or would that, does it have to be like diving and holding your breath?
For the spleen?
Yeah.
I think it's more of, I think it has to be extreme, like you need the pressure of the water on your body also helps to force blood out of the spleen. That's what they say. So I think you have to, I think it's more of exercising while holding your breath. So we saw that diving reflex, right? So it shuts down your heart. Right? And some people, you know, do this. Indigenous people have done this for a long time because we have fishermen or pearl divers that go down to pretty deep depths, like 20 meters, and fish while kind of like walking around on the ocean floor. Okay? And so we do have the ability to respond to diving and become more efficient at preserving the oxygenated rich blood. Okay, so it filters blood. It also destroys any microbes, right? So any microorganisms that make it into the blood, and so this is due to macrophages and lymphocytes. There's actually another type of cell that they're going to talk about on the video, which are called dendritic cells. And they're called dendritic cells. They're similar to macrophages in their function, but they are more branched. So dendrites means to branch, and so dendritic cells are just phagocytes as well as macrophages. Okay, so this would be the white pulp. So the red pulp and the white pulp. We can see the difference histologically between where what's going on in each area. Okay. So in a diagram of the spleen, you can see that there is a lot of blood supply. So the blood goes and then it kind of filters through that reticular connective tissue, comes in, and then it's picked up by veins that then carry the blood out. And then we see here that it's got connective tissue on the outside. And then this would be the red pulp, where we have cells that are breaking down red blood cells, macrophages, for example. And then the white pulp would be like little islands in this space where there's lots of lymphocytes that are responsible for detecting and attacking microorganisms. So we only have one spleen. So it is not an organ that is, we don't have two of it. It's not a paired organ, just one. Okay, so the other thing about our lymphatic system is that we have it in mucus places that are rich in mucus epithelial tissue. And so this stands for mucosa, mucosa associated, there we go, associated lymphatic tissue. And this is diffuse and widely distributed underneath the mucosa, the mucous membrane. So this occurs in places where we have the mucous membrane, like the bronchi, in the stomach, where we use mucus to protect the stomach lining from acid, and in the small intestine, and the appendix is also malt, considered to be malt. And so in the small intestine, these are the Peyer's patches. So when you think about it, these areas are in areas where there could be lots of microorganisms. So we could breathe in like yeast, we could bring in fungi, we could breathe in bacteria, right? We could digest. So when we eat stuff, our food has a lot of bacteria on it, right? So we need to be able to protect it from it, and specifically with the small intestine. And in the appendix is actually off the large intestine. And the thing about the large intestine is that it has good bacteria in it, and it can also have bad bacteria in it. So we can talk about the appendix has symbiotic bacteria. That are good, right? But the appendix protects us from bad bacterial infections. Right. So we have lots of good bacteria. And one of the things that the good bacteria does in our large intestine is that it produces vitamin K. So we have a strain of E. coli in our bacteria that produces vitamin K, and that vitamin K can be reabsorbed. And vitamin K is actually really important for blood clotting. And so if we don't have those symbiotic bacteria, oftentimes our blood will not clot properly, which is why when babies are first born, they get a shot of vitamin K in case they have to do surgery on them. And so they want their blood to clot. And when babies are first born, they lack the good bacteria, and the good bacteria actually has to colonize their digestive tract when they're first born through like breastfeeding and like putting stuff in their mouths that's got bacteria on it and that kind of stuff. Okay. So the appendix protects against the bad bacteria, but obviously the appendix can sometimes get infected itself, and then it can rupture, and then you get substances getting into your abdominal cavity and creating a widespread infection, which is very bad. And I'm going to show you a video of that too. Okay, so if we look at, this is your small intestine, and you can see the shiny part of it is the mucous membrane, and these little pockets, this is the malt, right? So this would be the mucus-associated lymphatic tissue right there. Okay, so this can sometimes, when we talk about it in the digestive tract, okay, this can sometimes be related to inflammatory bowel syndrome. And one example of inflammatory bowel syndrome is Crohn's disease, right? And so what this means is that you get chronic inflammation of the digestive system, and that can actually lead to bleeding. So you can become anemic, you can get a lot of blood loss, you'll have blood in your stools. And then it also can lead to rapid weight loss. So like I had a friend who just started losing weight, just couldn't, no matter what he ate, he lost weight, and he also had blood in his feces. And that means that their digestive system is actually being attacked by white blood cells. So this is an example of an autoimmune disease. So we talked about autoimmune diseases in the joints, as in like rheumatoid arthritis. So this is another example of an autoimmune disease. So So I'm going to show you a video. The first video is a video that talks about, in more detail than you need to know, immunology in the gut. And then the second video just shows surgery of an appendectomy. So when they talk about ILs, they're talking about interleukins. And so we talked about how white blood cells are able to communicate. And so an IL is just an interleukin, which allows the B cells and the T cells to communicate with one another. So this is the small intestine, and you can see that there's villi here, and then they're showing nutrients as well as microorganisms floating by.
The human gut can be the scene for devastating conditions such as inflammatory bowel disease, which arises through an improperly controlled immune response. The gut is often the body's first point of contact with microbes. Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients. However, these structures and the underlying tissues also host the body's largest population of immune cells. Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialized phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell, or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23. Effector T cells also appear on the scene, and these coordinate an escalation of the immune response by secreting their own inflammatory molecules, tumor necrosis factor alpha, interferon gamma, and IL-17. Soon after the effector T cells have arrived, a voracious phagocyte called a neutrophil is recruited. Neutrophils are critical for the clearance of bacteria. One weapon in the neutrophil armory is the ability to undergo a dramatic form of self-destruction called mitosis. This leaves behind a jumble of DNA saturated with enzymes called a neutrophil extracellular trap. Although this can effectively destroy bacterial invaders and plug any breaches in the epithelial wall, it also causes collateral damage to tissues. Slowly the tide begins to turn, and the bacterial invasion is repulsed. Any remaining neutrophils die off via apoptosis, a non-inflammatory form of cell death, and are cleared by macrophages. Epithelial integrity is restored by replacement of any damaged cells with new ones from the intestinal crypts. Finally, Tregs are recruited once again to calm the immune response. Targeting the molecules involved in gut pathology is leading to effective therapies for inflammatory bowel disease.
Okay, so in people that have celiacs, what is the body responding to? Does anybody know? Celiac disease? Gluten, right? So you saw those dendritic cells like coming up and sampling, right? And they could also sample pieces of food. And so if the gluten protein is present, then it can cause an immune response, right? An autoimmune response where it'll essentially damage those cells. And you notice that when the inflammation occurred, it could actually lead to the death of the cells lining the small intestine, and they lose their cilia in particular. And so they're not able to absorb nutrients. So gluten intolerance can cause that type of chronic inflammation as well. Are there any questions about that idea? Okay. So the immune system, the lymphatic system, and the digestive system. Do you remember what type of nutrient is absorbed across the small intestine and then is picked up by the lymphatic system rather than the circulatory system? Was it carbohydrates, nucleic acids, proteins, or lipids? Lipids, right? So lipids, remember, can just actually move right through the small intestine, across the small intestine, and they're picked up by the lymphatic tissue or the lymphatic vessels and transported in the lymph. Okay, and that's important because that means that it won't clog up the circulatory vessels. Okay, so the last video we're going to watch is the appendectomy. And then you're going to see.
This video contains graphic footage of a real surgery and is meant for educational purposes. Viewer discretion is advised. Hello, I'm Carl Lowe Jr., and I'm going to walk you through a live laparoscopic appendectomy for a ruptured appendix. We'll start by injecting some local anesthesia, make an incision in the skin, and then insert this trocar. We'll pass the scope down the trocar so that we can look as we go through all the layers of the abdominal wall. Once we get through the abdominal wall into the abdominal cavity, we hook up our insufflation tubing and inflate the abdomen with gas. We'll look inside, and you can see that there's quite a bit of inflammation there in the right lower quadrant of the abdomen where I'm pressing. The appendix will be there. We'll insert 2 additional trocars into the body, insert our instruments, and now we can begin our work. We need to peel this fatty omentum off of the appendix. You can see the tip of the appendix coming into view.
So the omentum is just connective tissue that holds
And
the
we want to
appendix in
just
place.
dissect out the appendix and mobilize it. We're going to push it over to the right of the screen.
Cutting here, they just.
You can see that murky fluid
They're just cutting the connective tissue
Behind the
and getting it
appendix.
separated from the rest of the tissues.
indicating this is a serious infection. The liver is coming into view there. We continue to tease some of that fatty omentum off of the appendix. And as we sweep more of the omentum off to the right, we see there's a lot of pus around this appendix, indicating that it has ruptured. We'll sweep the appendix over to the left of the screen, and now you can see where it has ruptured. Just below my grasper, you can see the rupture in the appendix. And you can see that the tissue has this black appearance where it's necrotic from the infection. We're going to place this stapling device just below the rupture of the appendix so we can staple off and divide the appendix. This device is nice because it will divide the appendix but then leave a row of staples along the appendiceal stump at the colon. We fire the device now and let it go, and now you can see the appendix is divided off of the colon with a row of staples on it. And that second firing of the stapler is dividing the blood supply to the appendix. We'll put this appendix in a bag and just set it off to the side for now. We'll take it out at the end of the case. We go back and look at our staple lines just to make sure that the appendiceal stump is securely closed and there's no leakage from the colon there. And we also want to make sure there's no bleeding. Everything looks good here. Sometimes people wonder what happens to these staples. They don't dissolve, they're metal staples, but they're also too small to set off any metal detectors. We don't ever have to go back and take them out, and they don't hurt. We're going to swing down now and look into the pelvis. Sometimes with ruptured appendicitis, pus can develop in the pelvis, and sure enough, it has. So we're going to suck out all this murky fluid, and you can see there's some white pus in there too. We're sucking all that out as well. We're going to clean all this murky fluid and pus out of the pelvis. Now, I don't want any more pus to accumulate in the pelvis, so I'm going to put a drain down in the pelvis. And we're going to bring this drain out through the skin and leave it in for 4 days and take it out after that. Now that the operation is completed, we'll pull the appendix out and just take it right out through the incision here. And there's the appendix. We'll take our drainage tube and hook that to bulb suction to suck out any other fluid that tries to accumulate in the pelvis, and then we'll close our skin incisions. This patient felt much better after getting that inflamed appendix out. He spent 4 days in the hospital, and on that day I took out this drainage tube, and he went home in great condition. Thank you for your interest in this surgery.
Okay.
Full anesthesia. This video.
Okay, so I'm gonna turn the light back on, and we'll just take a look at the review sheet. Does everybody have a copy of the review sheet with them? So remember that the final exam is going to be on Monday at 10 o'clock in here. Okay, so if you, well, actually, this might be my last one. So let me just look at it. Okay. I just want to make sure that we went over everything on the list. well, I didn't specify too much lymphatic system. So everything on the lymphatic system is fair game, but we talked about the function, the different functions of the lymphatic system, and then the capillaries and how there's no pump, right? They're blind-ended vessels. They have valves in them, and then the different lymphatic organs. Okay, let me just look at that. Does anybody have any questions about the review sheet? About the essay questions? Okay, so remember that your lab practical is on Friday. So I'm hoping, I want to start that at 10:30 again. So if you could come up at 10, but I'm not going to start till 10:30. Okay, and it is not over the bones, but everything since we did the bones in lab and joints, not over the joints. Okay, so I will see you guys on Friday.
So about the lab, you said everything but the bones and the joints, so it'd be like the heart.
And blood first. So we did like hematocrit, a blood slide, we did blood typing and that kind of stuff. So I think that's the first thing we did. We did blood, we did heart, we did blood vessels. Yep.
So that'll be about it.
Yep.
Blood, heart, blood vessels, slides, and stuff. So
Hematocrit analysis, blood typing.
So to make up the lab that I missed, do you want me to do that?
I can't do the diving reflex. I don't have time. So I gave you the handout.
Yeah, I got that.
Okay, and so just write on that handout, you can fill out whatever you can. So you can label, did you get the blood vessels? You want to label the blood vessels, and then you want to fill out that sheet.
Yeah, I got that.
You got that. You didn't get. Oh, I don't. And did you want to take this quiz?
Oh, God, I didn't even. I haven't
Okay.
had time to look at it.
Okay. So we're going to drop below quiz 4.
Okay.
You know, if you follow me downstairs, I can get you the diagrams.
Yeah, cool. I'll do that.
When can I take that last quiz?